Hello, I'm Miss Ginsburg with KnowAtom, and today we're going to be reading Inheriting Traits. This is a lab manual in Unit 5. Section 1, The Importance of Proteins. A whale named Isabella. In 1998, a group of scientists took pictures and genetic samples of a blue whale that was swimming near the Galapagos Islands. Then, in 2006, another team of scientists took pictures and genetic samples of a whale they saw off the coast of Chile. When they compared their samples, they realized it was the same whale. The scientists named this whale Isabella. Blue whales are the largest animals that have ever lived on Earth, larger even than any dinosaur. Blue whales can grow to be about 30.5 meters or 100 feet long and can weigh 160 tons. They are so large that their heart is the size of a Volkswagen Beetle and pumps 10 tons of blood through the whale's body. The size of the whale is a trait, a behavioral or physical characteristic of an organism. The reason scientists knew that Isabella was the same whale that was studied in both 1998 and 2006 was that the DNA matched in both samples. DNA stands for deoxyribonucleic acid. In a multicellular organism, such as Isabella, almost every cell in its body contains the same DNA. Scientists took samples from Isabella's skin cells and found that the DNA was the same in 1998 as it was in 2006. This confirmed for the scientists that the whale near the Galapagos Islands in 1998 and the whale off the coast of Chile in 2006 was Isabella, because an organism's DNA is unique to that organism. The blue whale is the largest animal to have ever lived on Earth. Studying DNA. Scientists who want to understand why an organism looks or behaves the way it does often begin with that organism's DNA. This is because DNA holds the instructions for making all of the proteins needed to keep the organism functioning. Remember that proteins are large, complex molecules that play many important roles in the body. Scientists have identified more than 100,000 proteins, which have a wide range of functions. Everything that takes place in an organism is either made of proteins or is a result of an action caused by a protein. For example, like humans, blue whales have an aorta, which is the main blood vessel. The aorta of the blue whale is so large that a human can crawl through. The aorta and all other blood vessels are made up of connective tissues. These connective tissues have highly elastic proteins that allow the tissues to regain their shape after they've been stretched or contracted. Elasticity is important for blood vessels because they are constantly stretched as blood flows through them. Hemoglobin is another complex protein that transports oxygen in blood. Keratin is a structural protein that makes up your hair and nails. Keratin also makes up the filter feeder system inside the mouths of blue whales that allow it to filter large amounts of water and food. Hormones are another group of proteins that regulate various functions. They do this by sending messages between cells to regulate the cell's life functions. Still, other proteins allow parts of cells to move and cause muscles to contract, so animals can move from place to place. Red blood cells carry the protein hemoglobin, which transports oxygen. Keratin is a structural protein that makes up people's hair and nails. It also makes up this whale's filter feeding system. All of your cells are constantly building and using proteins, and the instructions for making each protein are held in your DNA. This is why DNA has been called life's instruction manual. DNA is a polymer, a large molecule made up of many smaller molecules bonded together in a repeating chain-like pattern. These smaller molecules are called nucleotides. A nucleotide is itself made up of 3 types of molecules: a phosphate, a 5-carbon sugar, and a nitrogen-containing base. DNA gets its name from the sugar molecule, which is called deoxyribose. There are 4 nitrogen bases in DNA. adenine, thymine, guanine, and cytosine. DNA has two strands that wind around each other, which makes the DNA molecule twist like a spiral staircase. This shape is why DNA is called a double helix. The sugar and phosphate molecules make up the sides of the staircase, and the nitrogen bases pair up to form the rungs. The base pairs follow a predictable pattern. A always links to T, and C always links to G. Because the bases are complementary, if you know the sequence of nucleotides on one side, you know the sequence of nucleotides on the other side. The base pairs are held together by hydrogen bonds. DNA is a polymer made up of smaller molecules. DNA is universal to all living things. This means that the cells of every organism, including bacteria, plants, blue whales, and humans, store genetic information in molecules of DNA. However, individual organisms have unique sequences of nucleotides. Because Isabella, the blue whale, had a sequence of nucleotides unique to her, scientists could use her DNA to help identify her. DNA contains the instructions that tell each cell how to make every protein it needs. It does this with genes. A gene is a specific pattern of nucleotides that produces a specific protein or set of proteins, which in turn codes for a trait. In other words, each gene contains a specific structure of DNA that provides instructions for how to make one or a few related proteins. Different combinations of proteins determine an organism's traits. The environment also influences traits. We'll explore this more in the next unit. Genes are made up of hundreds or thousands of nucleotides, and multiple genes determine most traits in people. There are several steps between the gene and the protein. Making proteins is called protein synthesis. It is a multi-step process that involves many parts of the cell. In order to understand these steps, it is important to remember that the cell is a dynamic place and there is constant activity happening. Each organelle has a specific role to play in keeping the cell functioning properly. Remember that the nucleus is the command center because it holds the DNA. This means the nucleus is the place where all of the instructions for building proteins are held. Proteins aren't made in the nucleus, however, but on ribosomes, which are located in the cytoplasm of the cell. This means that the instructions in the DNA need to somehow get from the nucleus to the ribosomes. Genes hold the instructions for specific proteins. These proteins determine traits. Making a copy. The cells get the instructions from the DNA by making a copy of the sequence of the nucleotides stored in a gene. To begin this process, a part of the DNA first has to unwind and separate its two strands, unzipping down the middle. One strand can now be used as a template so that a copy of the instructions can be made and sent into the main part of the cell. The copy is made with a polymer that is similar to, but not exactly the same as DNA. This polymer is called RNA. Like DNA, RNA is made up of nucleotides that are made of sugar and phosphate molecules connected to bases. However, the sugar molecule in RNA is different from the sugar molecule in DNA. The RNA sugar molecule is called ribose, and it is how RNA gets its name, ribonucleic acid. And there is a small difference in the bases. Like DNA, RNA has guanine, adenine, and cytosine. However, RNA doesn't have thymine. Instead, it uses uracil. The final difference is that RNA is a single strand compared to DNA's double strand. A kind of RNA called messenger RNA, or mRNA, makes the copy of the gene. The nucleotides of mRNA pair up with their complements on the strand of DNA. C and G pair up, and A and U pair up. This process is called transcription, and it means to make a faithful copy of. Transcription produces a complementary strand of RNA that can be used to carry the instructions for building a particular protein out of the nucleus and into the cell. RNA is made up of nucleotides. mRNA makes a copy of DNA by pairing up its nucleotides. Building proteins. Transcription doesn't occur for the entire length of DNA. Instead, it happens for one gene at a time. Once transcription has occurred, the mRNA molecule leaves the nucleus through pores in the nuclear membrane. It can now move to where proteins are made, the ribosomes. Remember that in a eukaryotic cell, some ribosomes float freely in the cytoplasm, while others are attached to the rough endoplasmic reticulum. In prokaryotes, all of the ribosomes float freely throughout the cell. Both types of cells have thousands of ribosomes because of how important protein synthesis is. Each ribosome is made up of two smaller subunits, one small subunit and one larger one. The mRNA attaches to the ribosome between the two parts. Once the mRNA is attached, the assembly of the protein can begin. Proteins are made up of different combinations of molecules called amino acids. There are 20 amino acids, and the different ways that they form make up all of the different kinds of proteins. The sequence of amino acids is critical to the structure of each protein and therefore how it functions. The mRNA holds the instructions for that sequence because the mRNA is a copy of the gene from the DNA in the nucleus. The next step is translation, which means to put information into a different language. In the case of protein synthesis, the sequence of nucleotides need to be translated into the sequence of amino acid. The nucleotides aren't read individually. Instead, every three-letter base of an mRNA molecule called a codon is the code for one amino acid. For most amino acids, multiple codons exist that code for that amino acid. There are also start codons and stop codons that indicate where the gene begins and ends. The mRNA slides through the ribosomes so that each codon can be read and matched up with its specific amino acid. This requires another form of RNA called transfer RNA or tRNA. Like mRNA, tRNA is produced in the nucleus but functions in the cytoplasm. Special enzymes add the correct amino acid to one end of each tRNA molecule. The other end of the tRNA is called an anticodon because it is complementary to the codon that codes for that amino acid. For example, if a codon for a particular amino acid was CUU, the anticodon would be GAA. This allows it to bind to the codon so it can deposit its amino acid. Once the amino acid has been deposited with its codon, the mRNA slides through the ribosomes so the next codon can be read and an amino acid deposited by this codon's tRNA. As soon as the tRNA molecule has deposited its amino acid, it then detaches from the ribosome so it can get another amino acid. This process continues until the entire sequence of nucleotides has been translated into amino acids, which join together. When the stop codon is reached, there is a long chain of amino acids. This chain is called a polypeptide, and it forms part of or all of a protein molecule. The shape of a protein is very important because its shape is closely related to its function. For example, hemoglobin has a globe-like, rounded shape that allows it to travel easily inside blood vessels carrying oxygen. Some proteins are spiraled, while others are folded into coils. Proteins are constantly being built and disposed of. After we eat, proteins are broken down into amino acids. Those amino acids are then absorbed and used to make other proteins. mRNA is translated into amino acids. The structures of hemoglobin is essential to its function. Section 2, Passing on Traits. The journey of a sea turtle. Every year, the leatherback sea turtle travels up to 16,093 kilometers, or 10,000 miles, and crosses the entire Pacific Ocean in search of jellyfish, which is its primary diet. It then returns to tropical waters to nest and breed. The leatherback is the largest of all sea turtles. An adult can weigh between 227 and 907 kilograms, or 502,000 pounds. The male sea turtle never leaves the ocean, while the female comes on land to lay her eggs in nests on sandy beaches. Once she lays her eggs, the female sea turtle leaves them and returns to the ocean. The temperature of the nest determines whether the eggs will become male or female turtles. If temperatures are between 28 and 29 degrees Celsius, or 83 to 85 degrees Fahrenheit, the eggs will develop into a mix of male and female turtles. Temperatures warmer than this produce females, while temperatures lower than this produce males. Once the baby turtles hatch, they are on their own to make their way to the ocean. If they survive the journey, they will grow, becoming larger as they turn into adults. This is a leatherback sea turtle. These baby leatherback sea turtles are making their way to the ocean. Cell division and growth. As baby sea turtles grow and turn into adults, their cells aren't growing bigger and bigger. Instead, they grow because their cells are duplicating. They do this through cell division. Cell division refers to the splitting of a single cell into daughter cells, each with DNA from its parent cell. This is true of all multicellular organisms. For example, you started out as a single cell. That one cell divided, and then those daughter cells all divided, and then the daughter cells of those daughter cells divided. This continues until you reach adulthood, at which point your body will be made up of trillions of cells. Before a cell can divide, it grows until it is double in size. This growth is important because the cell also has to replicate its DNA. To replicate means to make a copy of. Remember how in protein synthesis, part of the DNA double helix unzips and separates so that a template can be used for making mRNA molecule. The entire DNA molecule can also unzip. This is because each strand of the double helix runs in opposite directions. At a certain point, the twisted, tightly packed double helix unwinds and separates its two strands, unzipping down the middle. Each strand serves as a template for a new strand of DNA molecules. The two new strands are both exact copies of the original DNA molecule because nucleotides are added wherever there are T's and C's wherever there are G's. DNA structure allows it to make exact copies of itself. Chromosomes. DNA replicates itself right before a cell gets ready to divide. Because of DNA replication, a copy of an organism's DNA gets passed along to the daughter cells. This is how all of your cells contain the same DNA. Right before a cell gets ready to divide, its DNA condenses into thread-like structures called chromosomes. Each chromosome is made up of a single DNA molecule, and it holds hundreds or thousands of genes on it. The genes are located on the chromosomes in a very specific way. Because of this, if scientists know where one gene is located, they can find it on anyone's chromosomes. Different kinds of organisms have different numbers and shapes of chromosomes. For example, prokaryotic cells don't have a nucleus, so their DNA is spread throughout the entire cell. Most bacteria have one or two circular chromosomes. Plants and animals have linear chromosomes that are arranged in pairs. Fruit flies have 4 pairs of chromosomes, while lobsters have 50 pairs of chromosomes. Right before the cell divides, its DNA becomes visible in the chromosomes. In humans, there are 23 pairs of chromosomes found in the nucleus of each of your cells, except for red blood cells, which don't have a nucleus. Chromosomes are in pairs because one chromosome is inherited from each parent. The first 22 chromosome pairs are the autosomes, which are chromosomes that everyone has. They are numbered according to size. The gender-specific sex chromosomes are the final chromosome pair. Biological females have an XX combination, and biological males have an XY combination. Both chromosomes in the chromosome pair contain genes that code for the same proteins. These are called alleles. An allele is a form of the same gene that has small differences in the sequence of DNA bases. For example, one allele might have the instructions for proteins that would result in blue eyes, while another allele might have the instructions for proteins that would result in brown eyes. Each parent contributes one allele. After DNA replicates, there are two of each chromosome. The identical chromosomes are called sister chromatids, and they are linked together by the centromere. Once a cell has replicated its DNA, it gets ready to divide so it can pass along its genetic information to its offspring. There are several reasons why cells divide. One important reason is growth. The more cells an organism has, the larger that organism is. Cell division also allows cells to repair damaged cells or replace dead cells. For example, human skin cells constantly divide so they can replace damaged or dead skin cells. Muscle cells also divide frequently to replace cells damaged by exercise or injury. Finally, cell division allows cells to reproduce. Reproduction is the ability of a mature organism to have offspring. Almost all cell division happens through a process called mitosis. Mitosis is a form of cell division that takes place in eukaryotic cells and results in two daughter cells with the same number and kind of chromosomes as the parent cell. There are 4 phases of mitosis. Phase 1, prophase. When the cell is ready to divide, DNA is wrapped even more tightly, and the chromosomes become visible. Each chromosome consists of the 2 sister chromatids formed during replication. The nuclear membrane breaks down, and the chromosomes are freed from the nucleus. Small fibers called spindle fibers begin to grow from the centrosomes on opposite sides of the cell and attach to each sister chromatid at its centromere. Phase 2, metaphase. During the metaphase, the chromosome pairs line up in the middle of the cell end to end. This formation ensures that each daughter cell will receive one copy of each chromosome. Phase 3, anaphase. The separation begins. The sister chromatids separate and the spindle fibers pull one complete set of an organism's chromosomes to each end of the cell. For humans, 46 chromosomes move to each end of the cell. Each side receives the same number and kind of chromosomes as the parent cell. Phase 4, telophase. During telophase, the chromosomes reach opposite ends of the cell. The nuclear membrane reassembles and encloses each cell's set of chromosomes in a nucleus. The chromosomes become indistinct again. After telophase, most cells undergo a process called cytokinesis, in which the rest of the cell splits apart, resulting in 2 separate daughter cells. So here are pictures of the prophase, the metaphase, anaphase, telophase, and cytokinesis. The result of mitosis is 2 daughter cells that are exact replicas of each other and the parent cell. In a multicellular eukaryotic organism, mitosis is how the organism grows. In a single-celled eukaryotic organism, mitosis is how the organism reproduces because the daughter cells are new organisms. This is a form of asexual reproduction because it requires only one parent. Asexual reproduction is the most ancient method for passing on genetic material. The passing on of traits from parents to offspring is called heredity. Most unicellular organisms reproduce asexually, although a few multicellular organisms also reproduce this way. In prokaryotic cells, which do not have a nucleus to hold their genetic material, cell division is similar to mitosis because the cell replicates its DNA, and the 2 copies split into 2 halves of a dividing cell. The result is 2 daughter cells that have the same genetic material as the parent cell. From a reproductive standpoint, there are certain advantages to asexual reproduction. Organisms do not need to expend time or energy searching for a mate, and all of an organism's genes are passed on to its children. It also can occur rapidly. For example, bacteria can divide several times per hour. However, there is very little genetic diversity in asexual reproduction. Because one parent passes along an exact genetic replica, the same genes are passed down from generation to generation. If one individual cannot overcome a type of disease or rapid climate change, then all other offspring from the same parent are equally vulnerable. Nature's answer to this problem of lack of diversity was sexual reproduction, which creates offspring which combine genetic information of a male and a female. The combination of genes ensures that each offspring has a different set of traits. So in asexual reproduction, you have the parent, and the offspring are identical to the parent. In sexual reproduction, you have two parents, a female parent and a male parent, and the offspring have unique traits. Humans and other complex organisms reproduce sexually through fertilization, the fusion of two gametes, cells that only contain half of an organism's chromosomes. When a gamete from a male joins with a gamete from a female, the two gametes create a new individual that has a complete set of chromosomes. Gametes are formed during a special kind of cell division called meiosis, which only happens when cells in sexually reproducing organisms prepare for reproduction. In meiosis, the cell divides twice, resulting in 4 daughter cells, each with half the chromosome numbers of the parent cell. Just as in mitosis, the chromosomes must duplicate before meiosis can take place. The same 4 phases of mitosis, prophase, metaphase, anaphase, and telophase, occur during both meiosis 1 and meiosis 2. However, there are a few key differences at various steps in the process. During prophase 1, the chromosome pairs line up. Because each chromosome has 2 sister chromatids, there are 4 chromatids close together. As they line up close together, the non-sister chromatids exchange genetic information. This is called crossing over, and it occurs at random places along the chromosomes. This is an important difference from mitosis because it results in chromosomes that have different genetic information. Fertilization is one way that organisms reproduce sexually. In crossing over, genetic information is exchanged between the non-sister chromatids. This results in chromosomes that have different genetic information from one another and the parent. During metaphase 1, the chromosome pairs line up at the equator side by side. The chromosome pairs separate and they are pulled toward different sides of the cell. The cell divides. There are now 2 new cells, and each cell has one chromosome from each chromosome pair. The chromosomes are duplicated, so each still has 2 sister chromatids. The 2 daughter cells get ready to divide again. In meiosis 2, the cells go through the same phases as in mitosis. Because there was no DNA replication, however, there are not 2 sets of chromosomes. In metaphase 2, the chromosomes in each of the 2 daughter cells line up in the middle of the cell end to end. The sister chromatids separate, and the nuclei of each daughter cell divides again. By the end of meiosis, there are 4 daughter cells. These daughter cells are called gametes because they have just half of the chromosomes as the parent cell, and they are genetically different from the parent cell and from each other. Meiosis produces 4 daughter cells, which are called gametes. Becoming a human. In humans, human males produce gametes called sperm that have 23 chromosomes. Human females produce gametes called eggs that also have 23 chromosomes. When a sperm and egg join together, they form a zygote, which is a single cell that contains 46 chromosomes and a complete, unique set of instructions in DNA. The zygote undergoes mitosis many times, dividing and forming new cells. Each new cell contains the same 46 chromosomes as the original cell. As more cells are formed, a complex process begins that will turn that group of identical cells into specialized cells, tissues, and organs. Eventually, those specialized cells will form a fully functioning organism, a baby. Some cells will reach out to others to form blood vessels. Groups of cells will bulge on both sides of the embryo to form the beginning of arms and legs. A precursor to a backbone will form, which will curl into a tail for a short time before disappearing. Scientists don't know exactly what causes cells to become specialized. However, since every cell has the same 46 chromosomes carrying the same genes, certain genes must somehow be turned on to tell the cell to build a particular protein, while other genes must be turned off. The process of turning genes on and off is known as gene regulation. It is an important part of normal human development. As the embryo is developing, genes are turned on and off in patterns that create the specific structures and function of brain cells, liver cells, muscle cells, and each of the trillions of cells found in the human body. Cells seem to know exactly where they are in the body, communicating with each other by sending chemical messages that trigger a reaction in neighboring cells to turn off that same gene. Through this process, each cell in your body is given a specific form and function that contributes to the functioning of your body overall. Differences between mitosis and meiosis. In mitosis, the nucleus divides once. In meiosis, the nucleus divides twice. In mitosis, 2 daughter cells are produced. In meiosis, 4 daughter cells are produced. In mitosis, daughter cells have the same number of chromosomes as parent cell. In meiosis, daughter cells have half the number of chromosomes as parent cell. In mitosis, daughter cells are genetically identical to each other and to the parent cell. In meiosis, daughter cells are genetically different from each other and to the parent cell. Section 3, Mutations. Drinking milk. In 2007, an international team of scientists went to Africa with a question: what caused the majority of Africans to be able to drink milk? This question may seem silly, but it is something that researchers have been investigating around the world. This is because not everyone can digest the primary sugar in milk, which is called lactose. A protein called lactase is responsible for the breaking down of lactose, cutting the lactose molecule in two. Babies are born with this protein functioning so that they can digest milk. However, research has shown that in our early ancestors, the genes that produce lactase somehow got switched off after babies stopped drinking their mother's milk. In some people today, their genes that code for lactase are still switched off as they become adults. These people are lactose intolerant, which means they cannot digest milk. However, in some populations around the world today, there are many people who are able to drink milk as adults because their bodies continue producing lactase. So what makes this possible? The answer is mutations, which are permanent changes to an individual's DNA. The scientists investigating their question gathered samples of DNA from people living in the region and looked for structural changes to the genes that coded for the protein lactase. Babies are born able to digest milk. However, some people lose that ability as they grow up. Structural changes to DNA. Mutations are relatively common. A mistake occurs every 100,000 or so nucleotides, resulting in about 120,000 mistakes each time a cell divides. The cell is able to repair most changes, but sometimes a change gets through. If a change occurs in an egg or sperm cell, it will be passed down to offspring. There are several ways mutations can happen. One way is when one base is exchanged for another, such as switching an A to a G. This can change a codon to one that codes for a different amino acid, which will affect the protein being built. Sometimes substitutions don't affect the production of a protein at all because the change to the codon results in a new codon that codes for the same amino acid. These are called neutral or silent mutations because they don't affect the organism. Other mutations occur when extra base pairs are added into a new place in the DNA, or when sections of DNA are removed from the DNA. Mutations can also occur in chromosomes, which can affect many genes. Environmental factors, including smoke and pollution, can cause some mutations. Sometimes mutations are harmful. For example, when there is a substitution in one of the genes that codes for hemoglobin, it alters a single amino acid in the protein. This results in a disorder called sickle cell anemia. Sickle cell disease causes some blood cells to become sticky, rigid, and shaped like crescent moons rather than flexible and donut shaped. Sickle shaped cells have trouble moving through small blood vessels, which can block blood flow and therefore oxygen to parts of the body. Other mutations can be beneficial. For example, the mutation that causes the body to continue producing lactase into adulthood benefits people who live in regions where they have access to milk and other dairy products. I learned a lot reading Inheriting Traits, and I hope that you did too. I'll see you tomorrow with the next one. Bye.